Hey everyone, my name is Dr. Stephen Hussey, and I am a chiropractor and functional medicine practitioner. I spent a lot of time because of my own health journey researching what causes heart disease and atherosclerosis. Today, I want to share with you some of that. So, we're going to talk about atherosclerosis and the effects of a ketogenic diet on exclusion zone water, which is one of my favorite topics because it's not talked about too much. And I think that it's very, very important. So, let's get started. First off, my disclosures. I don't really have any. I have a website that I sell my books off of. But other than that, don't really have any financial ties or anything to anybody in the industry. So I want to start off with this quote by Albert Georgi. And it says that science is built on the premise that nature answers intelligent questions intelligently. So if no answer exists, there must be something wrong with the question. And so oftentimes there is a lot of debate in this space about whether cholesterol or LDL cholesterol lipoproteins cause atherosclerosis. And there's a lot of debate. I think that things are unsettled. People don't know exactly if it does or not. And so I would argue that if no answer to this question exists, we can't settle on anything. Maybe there's something wrong with the question. And hopefully by the end of this, we'll get to a conclusion on whether or not there's something wrong with that question. So, usually, when we're looking to assess heart disease, we're taking things like lipid panels and looking at LDL, triglycerides, cholesterol, CAC scores. It's very focused on these fats in our blood, these lipoproteins in our blood, and different subsets of those and different formations of those, and really doubling down and assessing the state of the lipids in our blood. But I would suggest that there are other things we should be looking at, other things we should be concerned about, and we're going to make those relevant today. So this is a short video from Ivor Cummins at Low Carb Houston 2018, which he gave me permission to show you guys. And he mentioned something in here, and it was the first time on any of these talks that I heard someone mention this outer layer here. So let's see what he has to say.
Here it is shown in beautiful health. You can see the glycocalyx strands there, and there's an outer barrier area they've more recently discovered. And it keeps the red blood cells and platelets and other elements, as I said, away from the wall of your artery. Oh, here's one in a rotten state. So you've been chugging Coca-Cola or smoking cigarettes or whatever you've been doing, right? And as we can see here, the LDLs have much more access to the endothelium.
Right. So he's the first one I've heard mention this outer barrier. You can see it there. It's just kind of that big blue area sitting on top of the glycocalyx, which is those little blue hair-like things in the image. I'm not sure if you can see the cursor, but there's the glycocalyx. And so he's talking about this area here, this blue area. And you can see that in the studies that he's talking about, they've shown that the red blood cells are kept from the outside or kept on the inside and kept from touching the arterial wall. And then over here, he shows that one where this outer barrier has broken down, and that things in the middle of the lumen of the artery can now come into through that wall or through that barrier and connect with the arterial wall and the glycocalyx and things there. But there's something very wrong with these images, and as we go forward, you're going to see what's very wrong with these images, and it's going to clue us into maybe why LDL is and cholesterol in general is not the best thing to look at when we're assessing for risk of atherosclerosis. So now we got to talk about exclusion zone water, which will help explain this outer barrier situation that's happening in our arteries. And so fourth phase water and exclusion zone water, it's got a lot of different names. It's also called structured water. Has been researched by a lot of different scientists over the years. Albert Georgi, who gave us that quote at the beginning, is one of them. Also Gilbert Lang, and then more recently, Dr. Gerald Pollack at the University of Washington has written a few books on fourth phase of water, but that's his most popular one right there. And there's his publication on it. So what is fourth phase water? What is exclusion zone water? So water has these unique properties, unlike any other liquid, in that it can hold on to energy, it can absorb energy from the environment, more specifically radiant energy, which we'll talk more about. And when that happens, when water is sufficiently energized and it's next to a hydrophilic surface, a water-loving surface, then something interesting happens. So what happens is that one of the hydrogens on the water molecule is cleaved off, and the leftover oxygen and hydrogen team up with other oxygens and hydrogens, and they form these hexagonal shapes. They kind of bind together and form these hexagonal shapes, and then those hexagonal shapes, bind to each other, and they form like this, kind of like a fence posting, I think, or like a fence panel, I think, um, uh, set up like that, and then they stack on top of each other next to this hydrophilic surface, and it creates this area of what we call easy water. It's fourth phase water because it's not solid, it's not liquid, it's not gas. It's kind of between solid and liquid. It's kind of like a gel state. And so it forms this next to hydrophilic surfaces. So that's what exclusion zone water is. Now, one of the other interesting things about it is that because the one of the hydrogens is cleaved off, and the oxygen molecule is bigger than the other hydrogen molecule that's still there, the oxygen molecule is very negative, electronegative. And so when we get this easy water, it's a very negative charged area. And then the leftover hydrogens that were cleaved off end up outside of it, and it creates a very positively charged area. So we get this energy gradient that actually drives flow of water, which we're going to talk about more as well. But just to illustrate that the easy water is a very electronegatively charged space because of this distribution of those the oxygens and the hydrogens in the formation of easy water. Now, the other interesting thing is that it's called exclusion zone water for a reason, or one of its names is exclusion zone water for a reason. And that is because when these planar, like these fence panel like planar things that are formed of easy water, when they stack up on each other, they don't stack up directly. The hexagonal shapes don't stack up directly lined up with each other. They're kind of offset. And so you can see this picture here on the right, the very top image, where the hexagonal structures are kind of offset a little bit because the charges kind of keep them that way. And so that creates a very small space for which anything could possibly get through. And it turns out that very few, if anything, can get through this space. So it's called exclusion zone water because nothing can really penetrate it. And the only thing that's been shown to really penetrate it is very small hydrated ions. And so, so when we have this barrier here, it excludes anything else from going through it or touching the hydrophilic surface. It kind of protects it, which is very important for our discussion. So this is a video from a graduate student in Dr. Pollock's lab, and it just shows. So if you can see the cursor here, outside of this kind of tube thing, this is all like a gel. This is a hydrophilic gel, water, something that water likes to be next to. And so then in this tube here, it's filled with water, and then you can see the kind of the blurriness in the middle. These are little particles that they're floating around the water. But you can see that right here on the barrier, fourth phase water is forming, and it's kind of excluding the particles that are floating around the water. And so I'm going to hit play on this video, and you'll see that they're going to apply energy, radiant energy, to this system here, and more fourth phase water is going to form on the edge, and it's going to make the particles in the middle go toward, or here in the middle, get more narrow and more narrow of a space because it's excluding them. They can't get through the fourth phase water, so they end up more centralized. So let's play this and see what happens. So you can see more fourth phase water forming, but also I want you to note that the flow that's happening, the flow of this water is being created by the energy gradient of the fourth phase water itself. There's no pump or anything that's moving this water. It's moving more or less on its own because of the energy gradient. So the more energy they apply to that system, the more fourth phase water formed on the edge of the hydrophilic surface, and it narrowed the space where the water can flow, but it's also creating the flow because of the way that it forms energy gradient. So this is very relevant to us because of the roughly 50% of our blood that is plasma, 92% of that is water. So we're more or less, and our blood is more or less half water, a little bit less than half water. So there's potential here for this kind of mechanism to happen as long as there's a hydrophilic surface. And Dr. Pollock, this is a quote from his book, where he says that we also saw exclusion zones next to natural biological surfaces. They included vascular endothelia, regions of plant roots, and muscle. So vascular endothelia. So this The lining of our arteries is a hydrophilic surface, and our blood is nearly half water. And so you can see these pictures on the side here that are depicting that when we give enough charge to that water, enough radiant energy to our bodies, then that fourth phase water forms in the lining of the arteries, and we get a very negative space where the fourth phase water is, a positive space in the lumen of the artery, and this actually drives blood flow, which is a whole nother presentation. But it also is a protective layer for the lining of our arteries because it's exclusion zone water. It excludes things. And so this is a video also from Dr. Pollock's lab, one of his research students. And this is a chick embryo, the arteries of a chick embryo. And this is the flowing of blood after the heart has stopped beating. So it basically proves that fourth phase water does form on the lining of arteries in mammal or in animals, and that also it creates blood flow. So you'll see here, it kind of jumps around a little bit, but you'll see the blood flowing. So even after the heart has stopped beating, the blood continues to flow because there's still energy and there's still fourth phase water forming in those arteries. Okay. Now, here's another quote from Pollock's book, and he said he's talking about what's excluded from the fourth phase water. And he said, even red blood cells, several strains of bacteria, and ordinary dirt particles scraped from outside our laboratory were excluded. The protein albumin was excluded. Now, when I first read that, I thought it was kind of weird that he tagged on that sentence at the end, the protein albumin was excluded. But then I realized how significant it was because albumin is one of the smallest proteins that's carried around in our bloodstream. And so he's just noting that even this very, very small protein was excluded from the lining of the artery by this easy water. And so if we look at the size of albumin, it's 3.8 nanometers in diameter. So anything, we know at least that anything bigger than that would not be able to penetrate the easy water and contact the lining of the artery if we had intact, healthy, easy water lining our arteries. And so if we look at some things like a red blood cell, it's 6,000 to 8,000 nanometers, which is way too big. There's no way that that's going to penetrate the easy water, which the pictures that Ivor had in his presentation, they were depicting that very well, that the red blood cell was sitting on top of it, couldn't come through. An LDL molecule is 24 to 28 nanometers. So it's way too big. There's no way it's going to be able to penetrate easy water if easy water is healthy. HDL, also way too big. No way that it's going to penetrate that either. The only things that can penetrate them are these few hydrated ions, these ions that are fully saturated with water, sodium, potassium, and nitric oxide. And so those things, which makes sense because we know that nitric oxide is made in the lining of our artery. And so if there's this exclusion zone layer there, how does that nitric oxide get into the blood? Well, it's one of the few things that can squeeze through those gaps in the easy water and get through. But we're all told that LDL in high amounts is going to damage the lining of our artery. But if we have intact fourth phase water and we have a lot of LDL in there, they're never going to get past that exclusion zone if it's intact. Now, there's something called the Hofmeister series, which kind of gives us this linear size chart here. So it's the size of these hydrated ions, and magnesium is the biggest. NO3 is the smallest, and I guess the kind of the Goldilocks point there that the potassium and the chloride they can or the sodium and the sodium potassium is the one where it's kind of like on the edge, where those are like just small enough or just big enough, depending on their hydrated size, they can get through that easy water. But the calcium, magnesium are way too big in their hydrated size. So this Hofmeister series kind of gives us that size chart there. Now, if we look back at these pictures that from Ivor's presentation, we can see what's wrong. They're depicting that the LDLs, the little purple balls in there, are in the easy layer, and we've just shown that that is impossible. If easy water is formed, that the LDL, there's no way that it could get past there. Now, if it becomes damaged, if that layer of the easy water becomes damaged, then yes, those LDLs could sneak through there, and so could anything else that may be in the bloodstream. But we're going to see that it's not necessarily the LDLs, even then, that may be causing atherosclerosis or damage to the lining of an artery. We're going to see that it's the things that cause the damage to the easy water that are probably doing that. So, what builds easy water and what breaks it down? How does this happen? Well, building exclusion zone water is all about absorbing radiant energy from your environment. And so in Dr. Pollock's book, he noted that in his lab, they found that ultraviolet was the least effective, visible light was a little bit more effective, but the infrared light was the most effective, particularly the 3000 nanometer wavelength. And later they realized that the 3000 nanometer wavelength was the most strongly absorbed by water, which makes it the most effective at building easy water. And so in our environment, the source of infrared light is from the sun, the natural source, the one that our bodies are most used to is from the sun. There's UV light there too, but it also has infrared, and so people can also use infrared saunas and things like that. But that's just one way to build or to energize the water in our bodies. Also, direct contact with the earth. The earth gives off a lot of electromagnetic fields that are more compatible with our bodies. So here's another quote from Dr. Pollock's book. Lightning strikes so frequently around the earth that the earth's surface cannot dissipate the accumulating negative charge, leaving it electrically negative. Standing on the ground, your nose is about 200 volts more positive than your toes. And so we're absorbing that energy from the earth, and that's helping energize the water in our blood so that when it gets next to the hydrophilic surface of the artery, it can build fourth phase water and protect the artery and create blood flow. And so the thing that proves that this is happening to me is the studies we see with infrared sauna use. And the studies on infrared sauna and in people with heart failure, but also with artery health, are just phenomenal. And so it proves that when we apply infrared energy to the body, that this mechanism definitely happens. So here's one that says repeated sauna therapy increases arterial endothelial nitric oxide synthase expression and nitric oxide production in cardiomyopathic hamsters. And so you can see here that the sauna therapy at 34 weeks had way more nitric oxide, which means that that arterial lining is being protected so that those endothelia in the arterial lining can produce nitric oxide more readily, and then it can seep through the easy water, and we get way more nitric oxide production. Also, here's another one, this one done in humans with humans who already had heart disease. And they showed that just 2 weeks of sauna treatment lowered their blood pressure because when we're creating nitric oxide, that's creating vasodilation, and so that's increasing or decreasing the pressure in the arteries. And then also at the bottom, you can see that the percent flow mediated dilatation, which is again the dilation of the arteries and the flow coming through them, definitely increased after 2 weeks of sauna. And then also I found it interesting that the blood sugar went down, which was just a nice little bonus that's relevant to kind of our low carb community. But yeah, so these studies on infrared sauna definitely prove that when we apply the infrared light, it builds the easy water, protects our arteries, and creates healthier endothelia. So here's the conclusion. Repeated thermal therapy improves impaired endothelial function in patients with coronary risk factors, suggesting a preventative role for thermal therapy for atherosclerosis. So here's just a list of the things we can do to build easy water, light, especially from the sun, grounding, like we talked about, human touch. So humans, we give off compatible electromagnetic fields. So contact with other humans is very important. Vortexing, which is like the spiraling of water or kind of swirling of water in the presence of oxygen, is very important. That can actually energize the water. So then when it gets to a hydrophilic surface, it can form fourth phase water. Eating fresh food, because fresh food, like the water in the food that we eat, is also structured. But the longer it's been since it was picked or slaughtered or whatever, then the less easy water is going to be there. And then also, like we talked about, infrared sauna. So now we have to talk about the more important thing for our discussion is what breaks down easy water. And so here in Pollock's book, he says that suppose some electron-hungry process draws off some of the EZ's negative charge, leaving the released lattice unit devoid of its usual negativity. Issues of this nature could upset the default situation. So he's talking about electron hungry process, things that want electrons, things that demand an electron, and they'll go to any length to get it. They'll actually steal some of the electronegativity from the easy water and break it down. And so that electron hungry process in our situation, or in a human, or in most animals, is this thing we call free radicals, because they have an unpaired electron and they really want to be paired. They're very unstable when they're unpaired. And so they want to be paired, and they'll do anything to find a pair, including steal it from easy water or tissues, and cause damage when that happens. So free radicals are naturally made in the process of burning any fuel source, whether it's fat, carbs, or protein. Though we're going to see that some fuel sources are better than others. But also they can come from our environment. There's many things, chemicals and bacteria and things that can act like free radicals in our bodies too. These free radicals are normally neutralized by endogenous antioxidants like glutathione, which are made in our cells. They're highly reactive. Like I said, they want to be paired. And if they're unpaired, they'll do anything to get that pair, including steal it from easy water or tissue or whatever else. They have a very short half-life because they're so unstable and they want to be paired. They work real hard to get that pair. And they generate new free radicals by a chain reaction. This causes damage to cells, tissues, and exclusion zone water. So if we get a collection of free radicals that becomes this electron-hungry process, that could potentially compromise our easy water that's protecting our arteries. So this has been shown in the research. There's plenty of research on this, that oxidative stress definitely contributes to atherosclerosis. So we know that this abundance of free radicals, if we have it, this oxidative stress, if we have it, then it depletes the amount of antioxidants that our body makes. So this is one measuring here superoxide dismutase activity. And you can see that in the controls, they had way more superoxide dismutase available, whereas the people who had coronary artery disease, atherosclerosis of the coronary arteries, had less superoxide dismutase available, because their body had so many free radicals, so much oxidative stress, that it was depleting the antioxidants. So way less of it was available. And so this is what happens when we get the images like this, where the EZ starts to break down, is we have too much oxidative stress. And that's destroying the lining of the easy water, which is then allowing the same things that damage the easy water to then go and damage the glycocalyx, damage the endothelia, and then the body reacts and tries to repair that damage before it becomes a rupture or something. And we get the deposition of cholesterol and minerals and things like that, kind of acting like spackle. But the thing that we should be testing for as far as risk of atherosclerosis is not necessarily these LDLs that are just kind of innocent bystanders and getting damaged by that oxidative stress. It's the things that cause oxidative stress. Those are the things we should be really focusing on. And so I want to go through and talk about some of these sources of oxidative stress, and then what we can do dietarily to help us mitigate some of these things. So the first one is a glucose-based metabolism as a source of oxidative stress. So when our bodies need energy, they can burn three different things. They can burn glucose, they can burn fatty acids, and they can burn ketones. And all those things are converted to acetyl-CoA through various enzymatic reactions. And so then when it's acetyl-CoA, the body can take that and use oxygen through oxidative phosphorylation, and it can do respiration and make ATP. But which one is best? Which one is going to produce the least harm? Because as we burn these things, we're also going to make free radicals. It's kind of like the exhaust of the car. There's this waste product, and so which one of these is going to give us the least amount of waste products? Well, here's a few studies. This one's showing that oscillating glucose is more deleterious to endothelial function and oxidative stress than mean glucose. So this is talking about when blood sugars go up and down, and so that's more damaging to the lining of your artery, creates more oxidative stress than just mean glucose. And so the only thing that makes your blood glucose go up and down rapidly is when you're eating processed carbohydrates. That's the most damaging thing there. And so even if you're not diabetic, you're well controlled, eating processed carbohydrates is going to increase the amount that your blood sugar goes up and down, causing more damage, and than would fats or proteins. And here's another study down here that shows that a glucose challenge, so just giving the body glucose, stimulates reactive oxygen species generated by leukocytes. And so this is just showing that when we do glucose challenge, when we make blood sugar go up, this is just straight sugar. Then those situations, those foods create more reactive oxygen species, which are free radicals. In this case, they were looking at leukocytes. So I'm not a proponent that whole food carbohydrates in small amounts are damaging, but it's very clear that when we get foods that raise your blood sugar quickly and very high, that they create more oxidative stress than say fats or proteins. So here's a few studies that give us the opposite side of things, looking at ketones. So here's one: beta hydroxybutyrate elicits favorable mitochondrial changes in skeletal muscle. And so beneficial mitochondrial changes evident reduce H2O2, which is a type of free radical. Furthermore, muscle from rats and sustained ketosis similarly produce less H2O2 despite an increase in mitochondrial respiration. So despite more energy being created by the mitochondria, less free radicals were produced using beta hydroxybutyrate. And another one down here: antioxidant and anti-inflammatory activity of a ketogenic diet. This one just shows that beta hydroxybutyrate, the most studied ketone body, has been shown to reduce the production of reactive oxygen species, improving mitochondrial respiration. So if we're looking for a diet that can help us reduce the amount of free radicals made and increase antioxidants production, then a ketogenic diet can be that diet, as opposed to a glucose-based metabolism. So check that one off, and we'll move on to advanced glycation end products. So advanced glycation end products, this is what happens when something gets damaged by glucose. So in our bodies, this is what we're testing for when we look at an A1C, because we're looking at the percentage of red blood cells that have been glycosylated or damaged by glucose. So if our blood sugar is higher over an extended period of time, then more red blood cells are going to get damaged, and we get a higher percentage of red blood cells damaged in that A1C test. And so these advanced glycation end products act as free radicals. They are definitely something that can steal electrons and damage things. And so here we have the studies that show this: oxidative stress, AGE, and atherosclerosis. There is a definite evidence of oxidative stress and atherogenesis because numerous lipid oxidation end products, ALEs, or glycoxidation end products have been found to be increased in atherosclerotic lesions as compared to unaffected tissue. So they've actually found advanced glycation end products in atherosclerosis. When they dissect it out and study it, they find these in there. And the study at the bottom shows that AGEs can initiate and or propagate atherosclerotic lesions independently of diabetes or dyslipidemia. So if these are present, even if someone's not diabetic and has controlled blood sugars and normal lipids, then these AGEs can actually go in and start damaging the lining of arteries, because remember, they're damaging the easy water first. And that can contribute to the production of atherosclerosis on their own, independent of those things. So these things can be pretty nasty. And then here we can look at the effects of a low carbohydrate ketogenic diet versus a low glycemic index diet on glycemic control and type 2 diabetes. And so here, what they did was they put people on a low glycemic diet, which is the first section on the chart there, and then the low carb ketogenic diet. And you can see that when it comes to A1C, the number there at the end, the mean change, the low glycemic diet was good. It lowered the A1C about an average of 0.5 points, but the low carb ketogenic diet lowered it a full point lower than that. So if we're talking about lowering the risk of these advanced glycation end products causing oxidative stress or contributing to oxidative stress, then a low carb ketogenic diet is definitely more effective at lowering those, because we're seeing a decrease in the A1C, which means we're getting less damage by glucose to the red blood cells. So that's that one. We'll move on to endotoxemia. So what is endotoxemia? Endotoxemia is the presence of lipopolysaccharide or endotoxins, which is gram-negative bacteria in the blood. And so there's plenty of evidence here that endotoxemia uh, will definitely damage the lining of the artery and contribute to atherosclerosis. So here we have studies effects of low-dose endotoxemia on endothelial progenitor cells in humans, which endothelial progenitor cells are the cells that kind of initiate that atherosclerotic response. Endotoxemia is related to systemic inflammation and atherosclerosis in peritoneal dialysis patients. Potential role of endotoxin as a pro-inflammatory mediator of atherosclerosis, and association of endotoxemia with carotid atherosclerosis and cardiovascular disease. So these are just some of the studies that are available there, but it's pretty evident that the presence of these endotoxins in the blood can definitely act as free radicals, damage that exclusion zone water, and then therefore damage the lining of the artery, initiating that atherosclerotic response. And so where does endotoxemia come from? How do we get this? And so there are a few different places, and it all kind of has to do with the digestive tract. If we consider the mouth part of our digestive tract, which it is, one is that root canals, improperly done root canals, or even properly done root canals, you're leaving dead tissue in the body, and that dead tissue, that no matter how hard they try, they cannot clean out that tooth. As like the tooth part of the tooth that's left in there after the root canal, cannot clean it out, and so bacteria starts to fester in there. And so without a blood supply and without a nerve, because they drilled out the root of that tooth, the body can't fight that infection, and that endotoxic bacteria starts leaking through the tissues into the bloodstream, and that can give us endotoxemia. Um, and then also, here we have one systemic release of endotoxins induced by gentle mastication, association with periodontitis and severity. So basically, this is saying that if we have poor gum health, gingivitis, gum disease, then when we chew, that bacteria from the mouth is leaking into the bloodstream, because you think that blood is leaking out into the mouth there through the gums. The bacteria can leak the opposite way. So that can lead to this gram-negative bacteria, these endotoxins in our bloodstream. So the health of the mouth is very, very important. So, knowledgeable dentist that's aware of these types of things is a very important part of reducing our oxidative stress. But then there's the more famous one that contributes to endotoxemia, which is leaky gut. So when we have this, we have all this bacteria in our gut, and our gut is supposed to be sealed off all the way through. But if we are eating foods that poke holes in that gut, or we have we're getting toxins in our environment that are poking holes in that gut, whether it's molds or whatever it may be, and we get leaky gut, and things that should be kept in the intestines are leaking into the bloodstream, and a lot of times that is endotoxins, the LPS endotoxins, and so all these things contribute to endotoxemia, which, as we see, is a player in oxidative stress and therefore atherosclerosis. And so, what do we do about this? Well, first we make sure we don't have leaky gut, and we have healthy dental health. But also, cholesterol can help us out with this. So, lots of times people go on a ketogenic diet or a carnivore diet or whatever, and they see that their cholesterol goes way up, like we're seeing on this lipid panel here. And that happens for a few reasons. One is that the very process of making ketones, when you go low carb, forces the liver to make more cholesterol as well, because it's kind of the same pathway. It's like the same assembly line that diverges at the end. And so just by default, we get more cholesterol production. But also because we're eating lots of cholesterol, and our liver's not having to take fatty acids and make cholesterol and things from them, then the liver turns off the LDL receptors that would normally absorb that cholesterol back into the liver to do things with it. And it leaves those receptors off. And so that leaves the LDL in the blood. So we get higher amounts of LDL in the blood. But then also, especially on a carnivore diet, when people totally abstain from getting any plant fats have phytosterol, and animal fats have cholesterol. And so if we're abstaining from all those phytosterols, we're absorbing a lot more cholesterol, where usually they compete with each other. And so when this happens, it's usually, I don't believe it's necessarily a concern if cholesterol goes really high, as long as we have other markers in place. So on this blood work panel, I'd want the triglycerides to be a bit lower. I'd want the HDL to be a bit higher, but we'd also want to see that the inflammatory markers, like the few that I listed here, HSCRP, homocysteine, LPA. We want to see that those are all low. And as long as that's the case, then there's actually a benefit, I think, to having high cholesterol in some cases. And one of them is that can help us protect against various infections. So whether that's a viral infection or a bacterial infection, but also endotoxemia, which we're talking about here. And so here's a few studies that show the in vivo protection against endotoxin by plasma HDL. And low density or LDL receptor deficient mice are protected against lethal endotoxemia and severe gram-negative infections when they have more LDL available. So in that case, the receptor deficient mice could not reabsorb the LDL that stayed in the blood, and that helped protect them against the endotoxemia. And so then we have this study here that shows, you can see that the endotoxin recovery. So when they were taking blood samples and able to recover as many endotoxins, the recovery went down when they added various lipoproteins into the bloodstream. And you can see that as soon as they added, so ApoA, A1 wasn't that good at it, but ApoB was pretty good at it. But LDL and HDL were very good at it. As soon as they put them in there, it started going down. The amount of endotoxin started going down because these lipoproteins were kind of neutralizing the endotoxins in the blood, which can be very helpful for someone who is struggling with leaky gut or struggling with poor dental health. It could help them mitigate the risk of that while they get that stuff under control. And we hear this in the community, like Dave Feldman has called these people lean mass hyperresponders, where their LDL goes way up. And we also have various reports, and I hear oftentimes that these people have low rates of getting sick because these high amounts of LDL and lipoproteins in general they're helping prevent them from getting sick. So that's endotoxemia. So we'll move on to heavy metals now. So heavy metals are very common in our environments. So I just listed a few of them here: mercury, lead, aluminum, cadmium, arsenic. Some of those sources of them may be a little alarming or concerning to people. But the heavy metals in our body, or toxic metals in general, are definitely linked to oxidative stress. So much so that it's in textbooks, and I just put an example here that textbooks are teaching that exposure to these things are definitely linked to oxidative stress. And so you just want to look into your environment and figure out where you're getting exposed to these things and limit your exposure as best you can. But don't freak out about the ones you can't necessarily control, because that just adds to our stress. And when it needs to do that, just control the things you can, and look past the things you can't, and just kind of do the best you can. But these heavy metals are definitely associated with atherosclerosis. So here's a study where they looked at the levels of atherosclerosis in people and the intima media thickness of the carotid artery. And they showed that, as they kept increasing, or they kept seeing they did a hair mineral analysis. And so when people had these, the first 4 levels there, you could see that the amount of atherosclerosis they saw was generally about the same, a little bit higher in that third quintile there. But when they got to a certain point, when they got above 2.81, the amount of atherosclerosis jumped way up. It was 35% greater in the amount of people there in the carotid intima media thickness there. And so when they controlled for other co-founders, they found that it was a 31.9% greater amount of atherosclerosis in those people when they found this mercury in there. And so this mercury can come from dental fillings, it can come from eating high mercury fish, it can come from all kinds of places. But yeah, so there's definitely a definite association there. And so endogenous antioxidants are the best way we can fight against those heavy metals that have been in there. So here, this is a study that shows that a ketogenic diet increases mitochondrial glutathione levels, which glutathione is one of our main endogenous antioxidants. So the study here, you can see that the mitochondrial glutathione went up on a ketogenic diet versus the control. And so they also measured mitochondrial damage, which is an indication of high oxidative stress in the cells. And here they looked at it through H2O2 exposure. And so you can see that on the ketogenic diet versus the control, those white bars here, the control, the amount of DNA lesions was way higher. Ketogenic diet brought it down. And here again at this mark, it was way higher here, and ketogenic diet was keeping it low. So if we want to increase glutathione levels, increase antioxidant status, endogenous antioxidant status, like the antioxidants we make ourselves, then a ketogenic diet is one way that we can do that. So that's heavy metals. And then we'll go to BPA, which is a popular molecule. BPA is just a chemical produced in large quantities for use primarily in the production of plastics. And so I'll note here that BPA, lots of people, get BPA-free stuff. That doesn't mean that it's not full of these plasticizers, because they have to put something in it to make it plastic. So if they don't use BPA, they'll use BPS or BPZ or whatever else. And that's just BPA is just the most famous one. Everybody's like, oh, BPA is bad, so they just advertise things as BPA-free, but they have to put some kind of plasticizer in there to make it plastic. And BPA has been found in large amounts in humans, and just because of our contact with plastics. But it's not necessarily a free radical in itself, but it seems to create oxidative stress. And this study shows that the results of the present study are consistent with emerging evidence that human populations having higher levels of urinary BPA also have increased levels of oxidative stress markers and are prone to higher risk of cardiovascular diseases. So oxidative stress is what we're concerned about here, because it breaks down the easy layer. So having BPA there definitely contributes to that oxidative stress that breaks down the easy layer. So here's just a kind of a slew of studies. These are all done in rats and mice that show that the more BPA they're exposed to, the higher rates of atherosclerosis they have. And this one study with the male PRX humanized mice was pretty, the results were pretty striking. You can see here that what they did was they gave one group of mice the control diet, and then they gave the second group of mice the control diet with supplemented with BPA. So they added BPA to their food. And you can see that based on the controls here, the highlighted pink areas are more atherosclerosis. And so there's a little bit there, but there's way more in the BPA group here. And the same with this one here. There's a little bit of atherosclerosis, but there's way more with the BPA group. And so it's very clear that supplementing or exposing these mice to BPA definitely increases the rates at which they get atherosclerosis. And so this was a study done in humans. Circulating levels of BPA and phthalates are related to carotid atherosclerosis in the elderly. And you can see here that the phthalate metabolite MMP was related to atherosclerotic plaques in an inverted U-shaped manner independently of cardiovascular risk factors. So independent of all the other risk factors we assess for cardiovascular disease, these phthalates, these BPAs were highly associated with more atherosclerosis in these elderly people. So pretty concerning there. You want to make sure you're avoiding plastics whenever you can. So that's BPA, and that's it. But we have a bonus one, oxidized fats. So let's talk about those. So oxidized fats are just when the fats we eat or the fats in our bodies become damaged, and those things. And so this is usually the case with very unstable fats like vegetable oils. Those things can contribute to oxidative stress in a very very potent way. And so here we have this study where they looked at high cholesterol diet supplemented with sunflower seed oil, but not olive oil, stimulates lipid peroxidation. So lipid peroxidation is just damaged fats. The type of fats that we're talking about contribute to atherosclerosis. And so you can see here, they had a control in the chart. You can see control, and then the high cholesterol diet, then the high cholesterol plus sunflower oil, then the high cholesterol plus olive oil. And so malondialdehyde and diene conjugate here, the yellow and the blue, you can see that versus the high cholesterol diet, the high cholesterol plus the sunflower seed oil created more of this marker of lipid peroxidation or damaged fat. I went from 3.64 to 461, and here it went from 691 to 958. Same with the diene conjugate, 11 to 14, 1 to 2. So it just goes up. Whereas in the olive oil group, doesn't seem to do that as much. So that's a good sign. We just got to make sure that our olive oil is pure, because there's lots of olive oil brands out there that mix vegetable oil with olive oil. But then also I wanted to note that the glutathione In the high cholesterol group was around 30, but then when you do high cholesterol with sunflower seed oil, it goes way down. Didn't really change that much with olive oil, but you can see that these vegetable oils contribute to oxidative stress by damaging the lipid peroxide and also decreasing the amount of endogenous antioxidants around because it's getting used up to combat those free radicals. So we want to stay away from those vegetable oils that are easily oxidized. And here is a pretty cool study here that shows that the antioxidant capacity contributes to protection of ketone bodies. So we've seen this protection of ketone bodies against oxidative damage, reactive oxygen species, or oxidative stress. And so in conclusion, the present results show for the first time a direct reactive oxygen species scavenging capacity of ketone bodies, which is pretty cool, I think. That ketones, not only are they a better fuel source for us, but they can also scavenge up these free radicals, help us scavenge up. So if we have lots of ketones around, they can help us do that. So you can see that when they induce the oxidative stress in the control, this dark gray bar, the amount of oxidative stress went way up. And when you added various ketones, it kept it down. They were able to mitigate some of that oxidative stress, which is pretty cool. That's oxidized fats. So, how do we combat oxidative stress? Well, one way, as you can probably guess, I'm going to say, is a ketogenic diet. So, here's a study that shows that the effects of a short term ketogenic diet on redox status of human blood. And you can see that 14 days of a ketogenic diet increases antioxidant capacity in the blood of healthy subjects. And so, over here, the TAS is the total antioxidant status. And you can see that it definitely went up just after 14 days of a ketogenic diet. So if we want to increase our body's ability to combat oxidative stress and reduce oxidative stress, we need to pay attention to all the things I talked about: the endotoxemia, the glucose-based metabolism, the heavy metals, all those different things. But then we also can protect ourselves a little bit by having ketones around. So that can be super helpful. Now, just to kind of review a little bit, we have this hydrophilic surface that is the lining of our artery. And if we have water sufficiently energized enough that when it gets next to that surface, it can form what we call easy water or fourth phase water or structured water. And that acts like an exclusion zone, protecting the barrier of protecting the endothelial lining. But it also creates an energy gradient that creates blood flow. But if we get a source of or an electron hungry process from all these different things that can contribute to oxidative stress in our bodies, that can damage that easy layer. But it's kind of like the first line of defense. So if we have these things and we do things to protect our easy barrier and build our easy barrier, then we're protecting ourselves from this potential um, uh, oxidative stress that can damage the easy barrier. But if it gets bad enough, then yes, it will break down the easy enough that the things that are in the lumen of the artery can now contact the lining of the artery. And those same things that tore down the lining or the easy layer are the things that are now going to damage the artery. So it's not like these things damage the easy layer and then cholesterol can go and create atherosclerosis. It's there has to be damage first. And so those same things that create oxidative stress damage the lining of the artery, and then cholesterol comes in, or the body uses cholesterol and minerals and things like that to go in and kind of patch that up before it gets too bad. So we want to decrease our oxidative stress and also do things to build our easy layer. So in summary, water has the ability to hold energy and form easy water next to hydrophilic surfaces like our endothelium. This easy water is negative in charge and protects the endothelia by excluding blood elements, everything from red blood cells to LDLs to whatever else is in there, bacteria. Electron hungry molecules steal negative charge from EZ and leave the endothelia exposed. Oxidative stress is the electron hungry process and is contributed to by glucose based metabolism, advanced glycation end products, endotoxemia, heavy metals, BPA, and consumption of seed oils. The ketogenic diet has been shown to help protect the EZ water in our arteries from oxidative stress so that EZ can protect our arteries and prevent atherosclerosis. So, what does that lead us to as far as conclusions? One is that atherosclerosis is about much more than a lipid panel. And so, just taking a lipid panel or just looking at all the subfractions and everything of LDL is very important and very useful, can tell us a lot of things, but it's not the whole story. We want to look at markers of inflammation and oxidative stress as well. We also learned that LDL could be preventative in a sense because it can help us bind up endotoxins that may be there. And we learned that a ketogenic diet contributes to protection from atherosclerosis through these mechanisms, through the reduction of oxidative stress, therefore the protection of that easy layer that protects our arteries. And so because of that, we can see articles like this, lack of association, inverse association between LDL cholesterol and mortality in the elderly, where the review shows that the more LDL, the less mortality we have, because it's going to help us combat oxidative stress in some ways, and that eating more cholesterol, eating more low carb and higher fat is definitely going to help us reduce that oxidative stress, because the oxidative stress is not just causing damage to the lining of our artery. It can damage any tissue in the body, and it contributes to disease in multiple ways. And so anything we can do to reduce that is going to help reduce our ability to prevent disease, or increase our ability to prevent disease. And so then I just want to kind of leave with this note here, another cholesterol hypothesis, cholesterol as an antioxidant. So there's even some evidence that cholesterol itself can act like an antioxidant, which is just going to help us reduce that amount of oxidative stress. And so we'll go back to this quote here. Science is built on the premise that nature answers intelligent questions intelligently. So if no answer exists, there must be something wrong with the question. And you remember that our initial question was, does cholesterol or LDL cause atherosclerosis? And I believe that instead of focusing on whether or not it does, it's an important question, but instead of focusing so much on whether or not it does, we should look at what causes atherosclerosis and totally open up our view to anything that could potentially cause atherosclerosis, and stop being so myopically focused on the cholesterol and the LDL. So that's it. You can find me at resourceyourhealth.com or on social media there. And then also I have a new book out as of recently called Understanding the Heart. So check that out. Hope you enjoyed it. Thank you very much.